This conference will now be recorded.
All right. Well, good morning, everyone, and welcome to another EcoFoci seminar series. I am Deanna Krauser, co-lead of the seminar series with Heather Tabasola. This seminar is part of NOAA's EcoFoci biannual seminar series focused on the ecosystems of the North Pacific Ocean, Bering Sea, and U.S. Arctic to improve understanding of ecosystem dynamics and applications of that understanding to the management of living marine resources. Since 1986, the seminar has provided an opportunity for research scientists and practitioners to meet, present, and provoke conversation on subjects pertaining to fisheries oceanography or regional issues in Alaska's marine ecosystems. Visit the EcoFoci webpage for more information at www.ecofoci.noaa.gov. We sincerely thank you today for joining us today as we continue our all virtual series. Look for our speaker lineup via the One NOAA seminar series and on the NOAA PMEL calendar of events. Did you miss a seminar? Catch up on PMEL's YouTube page. It takes a few weeks to get these posted, but all seminars will be posted. Please check that your microphones are muted and you're not using video. And during the talk, please feel free to type your questions into the chat. We'll be monitoring these questions and we'll address them at the end of the talk. Today, I am pleased to introduce Michael Lomas. He is a senior research scientist and director of the National Center for Marine Algae and Microbiota at the Bigelow Laboratory of Ocean Sciences. As a marine biogeochemist, he has a broad interest in the role that phytoplankton diversity and physiology plays in mediating the key process of the biological carbon pump and associated macronutrient cycles. And with that, let's begin.
Wonderful. Thank you, everyone, for coming out today. It's great to see at least names on boxes of so many people I know. Come on. Why aren't we advancing? There we go. So today I'm going to talk about Synechococcus, a particular type of phytoplankton that's near and dear to my heart, and its abundance and biomass patterns in the Bering and the Chukchi Seas. And I hope by the end of this talk, you start to gain an appreciation for why it's important to understand whether or not this phytoplankton group is or is not actually in these regions. Before I get into the data, I really do need to thank a long list of people. In particular, I want to give a shout out to Pri, who's on, I saw her name on a box. Priscilla Lang, she works at the Department of Meteorology in Brazil, and has provided some of the slides for this talk. And so I wanted to specifically include her as a co-author tagline. A huge number of co-authors over the last 14 or so years, from Cal to Lisa to folks at UAF, Brad and Seth, and former postdocs of mine, other folks at PMEL, Phyllis, Dave, Carol, and some folks at NC State, plus a lot of other ones I haven't had time to list, as well as funding. So let's jump into the talk. Our subarctic and Arctic systems support high-value fisheries in large part because the food chains that exist in these systems efficiently utilize those nutrient inputs and efficiently channel that carbon and energy to higher trophic levels. Now, there's seasonal patterns and there's benthic systems, benthic productivities and water column productivities and the like. But the whole goal of the reason why these are so efficient is they are efficient at channeling energy. Part of the way in which that energy is efficiently channeled is because the chlorophyll, the phytoplankton that go through large seasonal amplitudes and reach very high levels, and therefore they can support high levels of productivity. This is very different than the open ocean systems where I spend the other half of my time doing research. When we look at these systems in the subarctic, when we have elevated chlorophyll, we tend to find very high percentages of large diatoms. So there's this nice positive relationship between total chlorophyll on the X and the fraction greater than 20 microns on the Y. going from large diatoms. Now, that being said, it's a line. So at the other end of the scale, where integrated chlorophyll is lower, you tend to have a greater dominance by either picophytoplankton or nanophytoplankton. And just for context, the Synechococcus data that I'll show you today, Synechococcus is a picophytoplankton. It's less than 2 microns. Okay. Come on. Come on. Why aren't we moving? There we go. But what we do know is that the Arctic is a dynamic changing system, right? It's warming rapidly, perhaps more rapidly than many other places on Earth. Yet we still really don't have a good understanding. Yet, that is to say, our understanding of what's going to be the impact on the lower trophic levels is still fairly limited. However, we do know that there's different patterns. So in the Chukchi Sea, when we use model data, the Northern Bering and Chukchi Seas, we see that the temperature anomaly tends to be more of a longer term trend of colder than average prior to the 70s or so, and then warmer than average in the 80s onward. Whereas in the Bering Sea, which is in the bottom panel there, you can see that it's more of an oscillation, at least during the record that we have that I show here, where the early 2000s are warmer than average and the later 2000s are colder than average. And the data I'm going to show today fall in the time frames outlined by these turquoise colored boxes. So our data from the Northern Bering and Chukchi Seas comes from the 2017, 18, 19 timeframe with one cruise in 2021, which is hot off the presses, as it were. And our data for the Bering Sea comes from 2008, 9, and 10. Now, why do we think there might be changes in the lower trophic levels as a result of these temperature patterns, both the global increase as well as these different patterns in increase? Well, the big reason is because other subarctic systems have shown that phytoplankton community size structure changes in relationship to temperature. So here's two sets of data from the published literature in the middle graph. Data from the east and the west North Atlantic in the range of 43 to 60 degrees north. You can see that on the y-axis, picophytoplankton, so small things like Synechococcus, as a percentage of the total phytoplankton biomass, increases positively with temperature on the x, so much so that by the time it reaches the maximum temperature of the data set, 80% or so of the population is these picophytoplankton. The data on the far right is from the Canadian High Arctic, greater than 70 degrees north, and I've outlined in color codes the real lines I want to draw your attention to. One is that for this 5-year period from 2004 to 2008, temperature was increasing. Chlorophyll in green here wasn't changing, so the biomass wasn't changing. But who made up the biomass was changing, and that's this blue line right here, which represents the picoplankton fraction. So their absolute abundance is increasing and changing, therefore, the size structure of the chlorophyll, which isn't changing. So phytoplankton niche models, right? So the armchair oceanographers or internet oceanographers, whatever we wish to call them these days, have tried to make predictions about what the world is going to look like, 10, 20, 100 years from now with regard to phytoplankton size structure. And one of these models by Flombaum et al. Suggests that the cyanobacteria Synechococcus will increase dramatically, most dramatically in the equator and the subtropics, but within the polar to subpolar regions, there seems to be some poleward shift, so extending their range closer to the geographic poles, and also, at least within the model constructs, potential for higher abundances. That's what all these sort of faded lines are, or all the different model runs that suggest in some cases there could actually be a lot more Synechococcus biomass shifted much further poleward. A more recent paper by Vicente suggests that, again, looking forward, that the magnitude of this response is dependent upon which climate warming scenario we enable, whether we use the RCP 2.6 or 8.5. But the key part is that it's these temperature-related changes seem to be specific to Synechococcus and similar organisms, not all the eukaryotes that do really well at driving energy and carbon into fish. So there's three questions I want to take and address today. One is the baseline pico phytoplankton community increasing or changing at all in the Bering and Chukchi seas as they warm? What are the potential broader implications of this rearrangement of the phytoplankton community size structure? And a recent addition is, can we assess or predict these changes at a larger scale so we can try to better understand how these systems are shifting? So let's start with, well, how do we actually quantify Synechococcus? So for the past 14 or so years, my lab has been getting samples or participating on cruises up in the Bering Sea to Chukchi Sea region. We have about 13 cruises worth of data now. And on the right, I just show various maps from science reports showing where we've sampled. So basically everywhere there's a dot, we have at least some samples at some season. So it's a pretty extensive data set that we can now start to interrogate and use for development of novel ideas, which I'll get to at the end. So how do we actually do this? Well, the use of a flow cytometer is, as I tell all my undergrads, really it's an expensive laser lit squirt gun. Expensive meaning it's more than my house mortgage. But this way this really works is you can see that we actually will inject, if I can get my pointer to work, that we run our sample through a nozzle and we intersect it with a laser beam. That laser will excite pigments in the cells. Those pigments will then fluoresce back some of that absorbed light. And so by looking at the spectra of the emitted light, and running it through gating schemes, we can basically chop up the population that we interrogate into different types of cells. So for example, in this bottom graphic here, all cells that absorb, all particles will scatter light. So if we take and try to separate those particles that emit chlorophyll fluorescence from those that don't, we can separate living cells from dead cells. Within the living cells, we can then go on and say, well, do they have phycoerythrin, which is an orange pigment? And if they do, we can gate them out and do further analyses on those. So for example, we can take and separate by size cyanobacteria, Synechococcus, which is small and phycoerythrin containing, from cryptophytes, which are large and phycoerythrin containing. For those that don't have phycoerythrin, we can then take and then also structure them by size. And the result being is that we can create lots of different populations within this pico nano community, phytoplankton community, very quickly. And as one would expect, the Bering Sea is a lot, or the Chukchi Sea as well, is a bit messier with lots of detrital particles kicking around. So what you can see here is bivariate plots of red fluorescence or chlorophyll fluorescence as a function of size on the left-hand graph, and then orange fluorescence or phycoerythrin and size on the right. And I've just drawn gates around how we decide who is who, right? So the red dots are Synechococcus because they are small and they have phycoerythrin. Cryptophytes here in the blue are bigger and also have phycoerythrin. And so that's just an example of what the base data looks like. We then take that data and now we have a count and we can relate that count to the volume we analyzed. And then we can create these interesting maps of what cell abundance is in different places. So I won't bore you with many of these because they start to blur together after a little while. But here's an example of what we commonly see the Southeast Bering Sea looking like. The top two graphs are section plots going on the map along the middle from Nunivak Island across the width of the shelf. The data are from April of 2008. Remember that 2008 was a cold year, and July of 2008. So April is on the left-hand side of these graphs, and July is on the right. And so the only things I really want to call your attention to are that Synechococcus is, as most phytoplankton, concentrated in the upper 20, 30 meters for the most part. They tend to be closer to shore. When we look at them in a spatial plot, which are the bottom sets of graphs, you can see that, interestingly, regardless of whether it's April or July, we don't see Synechococcus north of about 60 degrees in the southeast Bering Sea. You know, sometimes we don't see them south of 60 degrees north, but oftentimes that's where we do see them. And so we can see lots of spatial distributions. Now, just a sort of a general picture of what the Chukchi, Northern Bering and Chukchi Sea looks like from a seasonal point of view. Here's data from 2017, June, which was the Asgard 2017 cruises. It's here on the left, and September, the Arctic ice cruises on the right. Again, section plot up at the top. Again, showing that, you know, Synechococcus is closer to shore, just not right on the shore. When we look at it in a spatial plot, again, you know, if we look at just the Asgard data, that was encouraging because we didn't see it north of 60 degrees in the Bering Sea, and that continues into the North Bering and into the southern Chukchi. All these concentration of cell abundances are nice and low. What was surprising is that when we went and sampled late in the year, so this is our September cruise, the Arctic ice cruise, actually we saw massive amounts of Synechococcus. For the purposes of being consistent, I kept the scale the same between these two. So if you were to take and compare the Asgard cruise with the ice cruise, that's the net accumulation of Synechococcus from June to September. Because I don't like seeing a bunch of off-scale dots, I actually increased the scale by a factor of 10. So this is now up to about 15,000 cells. And so we can still start to see some of these northernmost samples actually coming on scale. But still, all of this region from Kotzebue Sound around Point Hope, Point Ley, sort of approaching in on Icy Cape, all still are off scale. So this was very surprising to us because we didn't see a seasonal signal in Synechococcus abundance in the southeast Bering Sea, but we're seeing a very clear seasonal signal, even though it's further north in the Chukchi Sea, Northern Bering and Chukchi Sea. So, well, why is that? Well, of course, as every phytoplankton person does, we just start correlating everything to everything, and it seems that temperature is the most interesting correlate with Synechococcus abundance. I guess if you were paying attention to the first few slides, you probably would have guessed that that's where I was going to end up, is that temperature warms, Synechococcus increases in abundance. On the left graph, that's all of our data. So the whole water column. On the right is just the surface 10 meters. There is a moderate difference in the ability to explain the data, but the reality is there's a very strong relationship between increasing temperatures and increasing Synechococcus abundance, even in the Bering and Chukchi Sea regions. But it's not all temperature. Temperature is just the dominant driver, it appears. So here I've taken and plotted the abundances of Synechococcus. They're the dots with the color scale as their abundance on salinity temperature space. And I've drawn in the warm coastal water and the warm shelf water masses that as defined by Seth Danielson in his 2020 paper. And so what you can see is that, as expected, there's no Synechococcus when the water is really cold, less than about 3 degrees or so, maybe even, a threshold closer to 5. And they see them most frequently in the lower salinity waters. Again, consistent with the fact that we find them closer to shore, and not, in the Anadyr Current and other areas offshore. The graph on the right is a dissolved inorganic nitrogen versus temperature plot. Again, the color of the dot is Synechococcus abundans. And as expected, you see all of your Synechococcus down here where it's warmest and most deplete in dissolved inorganic nitrogen. So we're suggesting as a hypothesis that Synechococcus may be a good biological proxy for coastal water influence. And when we see it further offshore, it may be sort of a tracer, in a sense, of offshore movement of these coastal waters. Although to really assess this, we need to find water with higher temperature and higher salinity values and see whether or not Synechococcus is there or not. But it does seem that it is growing quite healthily in these coastal waters in Alaska. But if you remember back to one of the earlier slides, I said, we have large diatoms on one end and really tiny Synechococcus on the other. And, it probably doesn't take a rocket scientist to see that it takes a lot of Synechococcus to be the same amount of carbon as a diatom. So how do we actually try to put these on the same scale now? And so we can actually get carbon from our flow cytometric data as well. And I won't go into the gory details of physics of light scattering, but the reality is that as cells go from being small to bigger, they change the way in which they scatter light. So a bigger cell scatters more light forward than a smaller cell does. And so what we can do then with our flow cytometric data is we can take and relate that forward scatter signal here on the X to the carbon content in particular cell types on the Y, and we get a really nice relationship across a very large range of cell carbon and cell scattered signal, both for field samples as well as cultured samples. So what we can do then is we can take our abundance maps and we can multiply them by this carbon conversion value, and now we can create carbon biomass maps associated with Synechococcus. And this is where it becomes really interesting, is because now we can start to compare them on the same units, carbon, as the diatoms. And so in this plot, we're looking at the integrated Synechococcus biomass, which shows an increasing seasonal amplitude over back-to-back warm years. So first start by looking at the leftmost graphs. These are the Asgard 2017 data and the Arctic Ice 2017 data. It's a surface plot. Now the Synechococcus is presented as carbon units. So this is their mass in carbon per meter squared. All of the color scales in all 5 plots are the same, so you can directly compare them all to each other. Starts off with very, very low carbon biomasses in the June of 2017. By the end of the summer, September timeframe, you can start to see the ingrowth of Synechococcus here in Kotzebue Sound is sort of working its way ever so slightly around Point Hope. When we look at 2018, interestingly enough, that all that biomass goes away, right? It resets itself almost to the same biomass level in spring of 2018 as it was in spring of 2017. We were fortunate enough with Phyllis's help to get some samples from the Healy 1801 cruise that summer, so it was a little bit earlier in the season than the Arctic Ice cruises. But what you can see is that there's a greater expansion of the spatial extent of high Synechococcus biomass. It's still in the same region, right? It's still around Kotzebue Sound. It's still going around Point Hope, maybe going a little bit further north now. But the spatial extent and the seasonal magnitude is increasing. We don't have data for the spring or June of 2019. For this talk, because I don't have any data, I'm going to assume that they reset back to the same levels like they did in 17 and 18. But now when we look at the end of the season, September Arctic Ice 2019 cruises, that spatial extent and biomass levels have yet further increased. So the seasonal amplitude is increasing from 2017 to 2019 as we have 3 years of continuously warm water. So what does that mean in terms relative to diatoms? Because diatoms are what fuel fish, or so we are trained as biological oceanographers that all fish are diatoms, I think is the exact quote from Cushing from 60 years ago. Diatom biomass in the spring has a huge range, anywhere from 0 to 4,000 millimoles carbon, but the average is about 21. That 21 is down in the bluish color on these scales. So in the spring, there's no blue, so diatoms are clearly more important. When we look at the end of season, the diatom biomass is much reduced. It's from 0 to 300 in its range, but it's about 18 millimoles carbon in the mean. Again, that's in the blue color, and you can see that everywhere where it's this sort of yellow, red, and peach color, Synechococcus is actually more abundant in a biomass framework than diatoms are. So to think now about what their relative contributions are, not just sort of a Synechococcus versus diatom war here, because there's lots of other phytoplankton, right? There's dinoflagellates, there's other flagellates, etc. So these two plots, I've looked at Synechococcus biomass as a percent of total phytoplankton on the Y and total phytoplankton on the X. And the color coding on the dots are temperature on the left and salinity on the right. And what you can see is that when you have really high biomass values, there's very few, if any, Synechococcus biomass hanging around, which is what we'd expect, right? Our blooms are dominated by diatoms. However, as you start to get to values that are only a factor of 2, maybe a factor of 3 below the maximum, Synechococcus biomass really starts to take off as a fraction of the total, oftentimes getting upwards of 40, 50%. And what you can see is in the left panel, it's the red dots. So it's the warmer temperatures or where they're most abundant, relatively speaking. And on the salinity plot, it's the yellows to greens, right? The lower salinity is where where they're most abundant. But thinking about phytoplankton and carbon, while works for me, it doesn't always work for those folks that measure chlorophyll. So let's try to sort of put this in chlorophyll space. These blue dashed lines represent the amount of phytoplankton carbon that one would be associated with 1 microgram chlorophyll, the long dashed line, and 2 micrograms of chlorophyll, the dash dot line. And it's in the same in both panels. And so you can see that once you get to about 2 down to or up to 2 micrograms chlorophyll, Synechococcus really starts to become a meaningfully important component of the community. Spending most of my time prior to work in the Arctic and the Sargasso Sea, we never see chlorophyll values this high. So to see Synechococcus becoming important at such a high chlorophyll was really interesting to me. But it is important to note that, much of the year, chlorophyll values are down in this range of 1 to 2 micrograms. And so Synechococcus is becoming a real important player in the biomass. So, the question is, well, has it always been there and we just didn't see it? Is it changing over time, increasing, decreasing? And to try to answer that question, I've tried to pull some data from the literature. And so there's a long list of caveats, like methods are all slightly different. They're all slightly different stations, etc. But I've tried to capture at least most of those caveats in what I'll talk about. So here we're looking at data in the Bering Sea. And again, at the top is that temperature anomaly plot that Phyllis put together all those years ago. There's data from Liu et al. From a cruise in 1999 in July, and shown by the gray dashed line here. That portion was a cold anomaly period. And what they found was that Synechococcus biomass was low, less than a millimole, so at the very, very low end, like we saw in the Chukchi Sea during June of 2017 and 18. Their cell abundances were all very low, 1,000 again at the very low end. Our Bering Sea data, which was compiled over 6 cruises, again shown by the gray dashed lines here between 2008 and 2010, show also during a cold portion of the record, show very similar maximum values. All of our biomass estimates were less than a millimole carbon per meter squared. I think there was only 1 or 2 samples where we had more than 1,000 cells per mil. So very similar to the Liu et al. Study from, roughly a decade earlier. Fortunately, we had the opportunity to get some samples from a cruise of opportunity in 2021, which we were all excited about because, after we sampled as part of the Bering Sea ecosystem study, a few years later, it started to warm up. Interestingly enough, 2021 was back as a below average temperature year. This is data from the NOAA ERSST version 5 data. Just chopped it down to not be overwhelming, but the black dot here is temperature in 21, and so it's a little bit lower than the mean in a sense. And interestingly enough, our Synechococcus biomass and abundance values were the same. So we really can't answer this question for the Bering Sea because by fate, dumb luck, choose your framework, we always seem to be sampling in cold years or cold periods. And so we don't really know what would have happened in, let's say, the early 2000s when there was a period of 4 or 5 years when it was warm, or in the late 2010s, again, when there's multiple years of warm water. So Bering Sea, we really can't answer that question of is it changing over time. The Chukchi Sea, however, I think we can answer that question. And, the punchline is it looks like it is, the Synechococcus abundance is increasing over time. So again, going through the literature, pulling out the data that I could find, one of the caveats is station. And so the data that I've compiled in these 2 graphs, the top graph is Synechococcus biomass, the bottom graph is temperature of the water at the time, all fall in the region of the Chukchi Sea that's bounded by this black box, whether it's our data or data from these other studies I was able to find. And so we can go back about to 2008 or so when Matt Cottrell and Dave Kirchman made some observations of Synechococcus. Biomass was very, very low. Temperatures were cold. There was a paper by Lee et al. 2010. Again, very, very low biomass, very low temperatures. Thankfully, Sam Laney and others made measurements during the IceScape program. Biomass is starting to get a little bit higher in its range, right? It's still sitting, values are at 0, but there's other values that are positive. Digging temperature out of the IceScape data has proved elusive to this point. So I don't know exactly what the temperature is, but it was, again, starting to show an increase just from the broader patterns that we see. The blue dots here are all of our the Arctic program data. So whether it's Asgard or Arctic ice, they're all here by these sort of mid-blue dots. And you can see that, again, while we do see in some stations no or very low contributions of Synechococcus biomass, the range over which they're extending is greatly enhanced relative to the data from a decade prior. And the temperature ranges are also much, much greater. So what happens? We go out in 2021 and we're all excited, except we now don't see nearly as large a range. Again, we see values close to 0 and there are positive values, right? We do see cases where their biomass is reasonable. It's just not nearly as high as during the Arctic program during 2017, 18, and 19. And so what we can't resolve yet is that because 2021 was an anomalous year? So this is a temperature anomaly plot looking at August 2021 minus subtracted from August 2020, showing that the Chukchi Sea region is in blue, which means it was colder than in 2020. So is this reduction in biomass because it's colder that year? Or is it because that cruise happened to be in November versus September? So there's a temporal pattern we can't resolve either, as well as it now dropping down in temperature relative to the longer term trend. But at least for the decade of 2008 to 2019, it seemed like Synechococcus abundance was increasing lockstep with increases in temperature in the Chukchi system. So what does that mean for the broader impacts? Well, first of all, smaller cells are less nutrient rich than diatoms, even if they are the same size as a diatom. So here's some culture data that we published a few years ago. On the top are data for diatom cultures grown at 2 degrees. The bottom 3 graphs are flagellates grown at 2 degrees. We don't yet have a Synechococcus polar isolate to do this with, so we're sort of using other non-diatoms as a proxy for what Synechococcus would taste like. And each of the graphs going from left to right are carbon content, nitrogen content in the middle, phosphorus content on the right. The black dashed line is actually the Redfield ratio. As a biogeochemist, that's something we're all sworn to uphold. In diatoms, all of the data fall above that dashed line, that Redfield ratio, which means they're all enriched in carbon, nitrogen, and phosphorus, much more so than flagellates are for each of those elements. So if a copepod were to sit there and eat 1,000 cubic micron cell, it's going to get a lot less nutrition out of a flagellate than a diatom. Again, thinking to what that means for biogeochemistry in these systems, the biology now is uncoupling in a way that the nitrogen and the phosphorus cycles in a way that's different than it had in the past when diatoms dominated, right? And so when we look at here on the far right side of this left-hand graph, this is the ratio of carbon to nitrogen, carbon to phosphorus, and nitrogen to phosphorus, where diatoms were abundant, again, all well below the Redfield ratio, especially for the carbon to phosphorus and nitrogen to phosphorus. And for the most part, we don't see that in flagellates. So again, the implications of biology doing elemental cycling differently will ripple through to the biogeochemistry of the system. And so we may need to, Cal and I may need to rethink how that works, what that means to the cycling in the system. So that's the Bering Sea, but the Chukchi Sea doesn't seem to show that same pattern. When we try to look at how elemental cycling of carbon and phosphorus in the Chukchi Sea behaves, when we see large fractions due to diatoms, all of these peach-colored and yellow and green dots, when diatoms are really abundant, they're all enriched in carbon relative to phosphorus. So the opposite of the case that we saw in the Bering Sea. And so, we don't know why this is. It could be a fundamentally different nutrient environment, but it could also be that the diatoms are adjusting to that new thermal regime. What I mean by that is that work that Jeff Krause and I published a couple of years ago. Even if diatoms don't change their size, even if they're still present in the system, their nutrient content will decrease as a function of temperature. That means that diatoms sticking around in a warm environment is less good for fish than it is diatoms sticking around in a cold environment, right? They start to look much more like flagellates, even though they're still a diatom. And so because our data in the Chukchi Sea was, in the middle of a multi-year period of warm temperature, was the physiology of the diatoms changing? And we don't have an answer for that. So it really sits as a hypothesis, but definitely will have an impact on all the higher trophic levels. As an example of what that means to higher trophic levels, and I apologize, these axes are backwards. These are because modelers always think in limiting elements first. But the reality is that global models predict that the Arctic regions will become deplete in phosphorus. There'll be a decrease in the phosphorus to carbon ratio in the phytoplankton community. And others have looked at zooplankton growth. Granted, Daphne is not the best proxy for Arctic zooplankton, but at least Daphne's growth is related inversely to food quality. As food quality goes down, so does its growth. So there's the potential for these ripple-on effects, whether they be changes in the type of community or the size structure, or just changes in the quality within a given diatom, that likely are to have negative impacts on higher trophic levels. So can we think about how to take this data set and expand it to make better predictions or just any predictions at all of larger scale patterns? This is work that we're actively ongoing under a newly funded JPSS project. Satellite retrievals here on the left have shown that in the, basically the 2000s, so 2000 to 2010, chlorophyll wasn't really changing much in the Arctic, but after 2010 or so, the chlorophyll started changing, right? And so there's a fundamental change in the driver of primary production. But we also know that somewhere in there's also changes in the underlying phytoplankton community composition that impacts chlorophyll. And so the goal for our JPSS project is to try to look at developing algorithms for Synechococcus prediction and improving our diatom algorithms. And this is the work that Pri and Jens Nielsen are leading. And it works from the base premise that phytoplankton of different types have different spectral absorption, and therefore you get differences in reflectance spectra based upon who is in the water column. And so we can take and use those differences in reflectance spectra and with temperature primarily to try to build these algorithms for Synechococcus abundance. And so these are quite literally data hot off the press. I think Pri sent these to me either Monday or Tuesday night. I forget exactly which night, but these are plots of our estimates on the y-axis of predicted, in this case, chlorophyll from the reflectance spectra and from the temperature in situ for the IceScape data, where we have that reflectance data, plotted as a function of our observations of direct extraction chlorophyll. And we can see that whether it's with our reflectance in a PCA model or reflectance, PCA model, and sea surface temperature, we get very similar levels of predictability, and they're good levels of predictability. One of the nice things about the IceScape data is we have lots of pigment data. One of the pigments is zeaxanthin, which is a proxy for, or which can be a proxy for cyanobacteria, of which Synechococcus is. And so we tried the same thing of using reflectance in a PCA model or reflectance and combined with a sea surface temperature model, trying to predict again on the Y and compare with our observations on the X for zeaxanthin concentration. And again, you can see that we actually do a pretty good job of predicting the zeaxanthin concentrations, and it's improved slightly when we use sea surface temperature. Again, consistent with our observations that temperature seems to be a dominant environmental driver. So with that, I just want to leave you a few take-home messages. One is that Synechococcus is not the trivial component of the subarctic community that we previously thought, at least during warm periods. Synechococcus biomass can be high both in absolute and relative terms. It seems to be, or the biomass is highest when it's warm, less saline, and nutrient-depleted coastal water conditions. At least within the Chukchi, where we had back-to-back years of warm or increasingly warm water temperatures, their net seasonal increase in biomass was growing, and their spatial extent was expanding. And our initial algorithm development attempts to actually try and back predict where Synechococcus may be are starting to look to be promising. But those are quite literally very new, so there's a lot of work yet to be done. on our prediction abilities. And so with that, I thank everyone for listening, and I'll stop sharing my screen and I'll take questions.
Thank you so much, Mike. I'll clap for everybody.
Clapping loses something when everybody's on mute. I get it.
And
Oh.
to do it for you. Calvin, of course, already has a question.
Yep.
My camera's being really slow, so I'm sorry about that. I may just turn it off.
Yep.
Go ahead, Deanna,
Yeah.
read the question.
Okay. Regarding Sienna cocoa
Is that better?
carbon content
Okay.
and HFC in summer 2018, 2019, what is the driving and dramatic increase across the Bering Strait?
That's an excellent question, Cal. I think in part it is, we haven't, sort of laid out how far from the U.S. side to the Russian side of the Bering Strait, the ACC extends and things like that. All right. Now let me share. Let me see if I can move that figure back because Cal's wants to see the picture again. There we go. All right. Come on. So yeah. No, I think part of no, too far. There we go. Get rid of this. So you're talking about like down here, Cal? Is that what you're talking about? Oh, Cal. Yeah. Now Cal's just being a punk. So I think part of this is just the slow expansion and the elastic coastal current sort of wrapping around here. Sorry, I'm trying to keep up with Cal's commentary and questions, but I think part of this is this whole region is warming a little bit, and so it's creating an environment that's much more conducive to Synechococcus growth. Is my hypothesis for now.
All right. Great question. Any other questions? All right, looks like we've got a question from Ryan. Hey, Mike, great presentation. What was the source of the satellite chlorophyll data showing the increase in the Arctic? OCCCI, Hermes, NASA?
Yeah, so that's the Lewis et al. 2020 paper. I am 90% sure, or 95% sure to be statistically accurate, that it was MODIS data, but I'd have to go back to the paper to actually refresh my memory of the exact source. So that data, because it covers such a long span, there's also a change in data sources from the early part to the latter part. And so whether or not some of that change is due to data source is a question. But assuming that it's not a data issue or things like that, it's that pattern, that flat chlorophyll to increasing chlorophyll while primary production was increasing, fundamentally suggests there was a change in phytoplankton physiology, right? It suggests that in the early part, they weren't accumulating their biomass, but they were continuing to grow. And in the latter part of that time record, they're now accumulating their biomass, but because the productivity rate hasn't changed, they're slowing down their growth rate. So there's some really interesting physiological adjustments that are just, hidden within satellite data that would be really worth exploring.
All right. Let's see if we can get one more question, and then we can close it up. Thank you so much for presenting, Mike. Oh, we got another comment from Calvin. Let's see, he says, new PACE satellite will present new opportunities for exploring phytostructure.
Yeah, no, absolutely. So I'm working with Antonio Menino and other members associated with the PACE science team to try to develop better phytoplankton structure algorithms. Um, Obviously there'll still be some regionality, but that's something we're trying to address is the regionality of these algorithms as well as just improved algorithms. So I absolutely agree. That's going to be a great opportunity when it gets here, and hopefully we're prepared to better understand the data.
Can you elaborate on what PACE is?
So PACE is what? The P is pre. It's the aerosol clouds, and I forget what the E is. It's a new satellite ocean color mission.
Oh, awesome. Okay.
It is hyperspectral versus multispectral, so it'll allow better resolution optically, which means that we'll be able to tell better similar phytoplankton groups from other phytoplankton groups because we just have that hyperspectral aspects of their absorption spectra and reflectance.
Great. All right, we got 2 more questions.
Thank you. Pre just wrote the ecosystem is the E.
Oh, there you go. A question from Libby. Do you know if there have been any changes in the zooplankton size spectrum? Thank you, Libby, for representing the Zoop team.
That is an awesome question. Isn't that what we're going to explore as part of our Arctic synthesis project? I honestly don't know, mostly because I haven't spent the time to look at the zooplankton data. But yes, Libby answered her own question and said yes. So, at least in the Bering Sea, we're seeing those changes, but we don't have good phytoplankton size structure necessarily to line up with the zooplankton size structure. So, but I definitely think now with a better view of phytoplankton community in the Chukchi, we should be able to take and start to explore, those connections and the impacts of size structure changes, as well as potential quality due to some of the other variables measured.
Oh, how exciting. All right, one more question from Emily. It sounds like there's some uncertainty in the influence of temperature versus timing of the samples. Do you have a hypothesis about the role of interannual variability versus seasonality?
Yeah, so the uncertainty there comes from the fact that in our 2021 cruise, which was the most recent data, was not at the same time of year, right? So it was, a month and a half or so after the Arctic EIS cruises. So that's the only reason why I can't separate it out is because I now have 2 variables changing. It's colder, so I would expect it to be lower, but it's also later, so I would also expect them to be reduced because of that. We can look at the interannual variability, but from the data we have, it does indeed show that their abundance, the net accumulation over the season is increasing from 2017, 18 to 19, and their spatial extent is increasing as well. So I think it's safe to say that, the interannual variability is at least as great as the seasonal changes of whether or not you're beginning or end of the fall season. But I think part 2, it's whether or not you have a series of years that are continually warming on each other, or if the system is resetting itself, at a much higher frequency, like what the Bering Sea did, right? It was, started back before 2000. It was roughly every year it was warm, then cold, and it moved to this pentadal structure of, roughly 5 years that were warm, roughly 5 years that were cold. Those ecologically, those have very different impacts on how phytoplankton structure assembles itself.
All right. We've got time for maybe one more question, and then we'll conclude. Let's see. This is cyanococcus. I'm struggling with this word.
You can just say syn.
Thank you. It is more abundant in higher temperatures, which is more straightforward, and in fresher waters, which I don't quite understand. Will you explain this a bit more?
Yeah. So, I think the salinity aspect is actually really, really interesting, because it's not a variable we generally associate with Synechococcus. It's mostly temperature, right? Whether it's, these global transects that have been done looking at Synechococcus abundance, temperature is a primary driver. My hypothesis is that, because Synechococcus actually does better in nutrient deplete waters, really the salinity is a sort of a covariate with low nutrient waters, which are coming, the Alaska coastal current and things like that. So that's my guess of why there's a relationship to salinity, that it just is really a mirrored reflection of the low nutrient environment in which it likes to exist, versus, the Anadir current, right? That's, cold and salty and nutrient rich, where Synechococcus just simply gets out competed.
Wonderful. Thank you so much for that question, Gakshu. I encourage everyone to reach out to Mike if you have any questions, but we're going to go ahead and close the meeting. We have meetings following this. Thank you so much for coming. It was a great talk. Thank you so much, Mike, for presenting. We'll go ahead and
thank you all for the opportunity to present the work that we have all been up to. I appreciate it.
Yeah, absolutely.
Cool. Have a wonderful day, everyone.
Hey, Mike, this is Cal.
Yep.
Have we stopped